Hello everyone, welcome back. Today I'm here with the sixth part of this question series. In this part I will discuss questions from plant physiology and plant economic botany. Let's dive in. The first question for today is, most of the leaves and flower buds open because of differential growth of their surface. This is known as, our options are thigmonesty, hyponesty, schismonesty और epinesty. So जो फ्लावर्स हैं और लीव्स बर्ड्स हैं वो ओपन होते हैं, वो किस तरह के मोमेंट्स की वजह से ओपन होते हैं? सो इट्स राइट आंसर इज एपिनेस्टी. एपी मीन्स अपॉन. एपिनेस्टी इज डिफरेंशियल ग्रोथ ऑफ द सरफेस ऑफ लीव्स एंड फ्लावर्स बर्ड्स कॉजिंग देम टू ओपन. और ये जो मोमेंट है ये डिफरेंट होती है फ्रॉम थिग्मोनेस्टी. जो थिग्मोनेस्टी मूवमेंट होती है इट इज़ नॉन डायरेक्शनल मूवमेंट. ये भी स्टिमुलस के लिए होती है. दिस इज नॉन डायरेक्शनल मीन्स इसकी कोई डायरेक्शन स्पेसिफिक नहीं होती है. Like we have response stimulus like touch and vibrations. In addition to this, we have some other types of nastic movements, like we have photonasty. Photo means light, so it is a bending or movement towards light. Then we have hyponasty. Hyponasty means जब growth upward direction में होती है, means it helps in closure of flowers. That we'll understand with the help of image. This is the image showing epinesty, that is bending of leaves. और ये जो conditions होती हैं ये arise होती हैं during disease condition. जब bacterial, fungal और viral infection होता है तो ये condition arise होती है, and mostly in flooding condition. And the hormone which is responsible for this epinesty is ethylene, which is a gaseous hormone. So this hormone is also responsible for this downward bending of leaves due to differential growth. Means जो upper leaf surface है वो ज़्यादा grow करता है as compared to the lower leaf surface. That leads to this bending or movement that is called epinesty, and this is responsible for opening of flowers and buds. So with the help of this image we can understand this is epinesty, means the upper leaf surface will grow more and leaf will bend downwards, while in case of hyponesty the lower surface will grow more as compared to the inner surface, and it leads to closure of leaves or upward growth of leaves. Then we have other type of movement that is called circumnutation, which is the bending of stem or soft stem towards some support. So hyponesty, when the growth is more on lower surface, that is called hyponesty. Petals show curvature on upper side and ultimately the flower becomes closed. So ये जो movement है, this is called hyponesty. While in case of epinesty, when growth is more on the upper surface, petal shows curvature on lower side and ultimately the flowers open. So the flower usually opens at high temperature and remains closed at low temperature. There are some flowers which show this type of movement. The next we have this nutation. This is again the growth of stem apices occur in zigzag manner. जब zigzag manner में growth होती है जो stem tips है तो उस condition को हम बोलते हैं nutation. It is because of the two sides of the stem apex alternatively grows more. Such growth moments are known as nutational moments. And in some plants that we can observe here, mostly the member of family Cucurbitaceae, इसमें जो nutational moments होती हैं that allow shoot apex to grow helically path in upward direction, like that is called circumnutation. In spirally growing stem, the tendrils, the region of greater growth passes gradually around the growing point, resulting in spiral coiling of the tendril or stem. So that growth is known as circumnutation, and it is found in the member of family Cucurbitaceae, and such movement are known as circumnutation. The next we have this thigmonesty. Thigmonesty is non-directional movement of plant in response to touch, where the plant part move in direction to physical contact with an object. On the other hand, schismonesty is a type of movement that is caused by mechanical stimuli like shock, touch or contact, leading to rapid movements in response to these triggers. Like the best example is Mimosa pudica. जिसके जो leaves हैं they become drooped when the leaves are touched by some stimulus, or we can say some touch or mechanical response like shock. The next question is, center of origin of wheat plant is, our options are Africa, South America, Southwest Asia and Brazil. So its right answer is Southwest Asia. And there was a region named as Fertile Crescent, and which include the area of parts of Iran, Iraq, Turkey, Syria, Lebanon, and Israel, and is considered as one of the oldest site for agriculture. तो यहाँ पे जो साउथ वेस्ट एशिया यहाँ पे ओरिजिन माना जाता है व्हीट प्लांट का, एंड द एग्जैक्ट लोकेशन इज़ कराकडेंग माउंटेन्स ऑफ टर्की, जहाँ पे जो हमारे पास मॉडर्न व्हीट है उसका ओरिजिन माना जाता है. If you talk about this wheat, it belongs to family grass family, that is Poaceae, or the genus that is Triticum, and it's common. This is the modern wheat or bread wheat, which is mostly grown or widely used throughout the world. और जो इसका origin है, that is considered to be in this region, that is Fertile Crescent, around 9600 BC. और जो Fertile Crescent है, it includes the area of or we can say part of Iran, Iraq, Turkey, Lebanon, Syria, or Israel. और if we talk about its production, तो इसकी जो production हुई है, that is 771 million tons produced in the year 2021. Nutritional value, it is a good source of carbohydrates, leading to a source of vegetable proteins in human food. It contains one protein, that is gluten, जो थोड़ा सा healthy नहीं होता है. The protein which is found in this wheat, that is gluten, it is not that much healthy, and it can cause certain diseases or discomforts in human body. And next comes center of origins. So this concept of center of origin was given by a Russian botanist or geneticist N.I. Vavilov. The name of the person was N.I. Vavilov, and he was a Russian botanist and geneticist. Based on his study on about 250,000 plant samples, which were gathered by a group of Russian botanists, including himself. He identified the following 8 centers of origin of cultivated plants, which include Chinese center, Indian center, Central Asiatic center, then we have Near Eastern center, Mediterranean center, Abyssinian center, South Mexican center, American center, and South American center. So these are the center of origins of different crops, and he divided the world's geographical area into these 8 centers of origins. The next comes origin of wheat. So the basic chromosome number in case of wheat is 7, and the diploid wheat is with chromosome number 14, that is Triticum monococcum. It is commonly known as the einkorn wheat. So it was crossed with a wild goat grass named as Agilops speltoides, whose genome was B. When they are crossed, they produce a diploid hybrid which was sterile. Then it leads to chromosomal aberrations or chromosomal doubling, और उसके बाद जो एक tetraploid plant हमारे पास आया, that is this one, whose genome is AAPB, and it is tetraploid wheat which include wheat like we have Triticum turgidum, जिसे Ammer wheat कहा जाता है, and its chromosome number 28. And there are some other tetraploid wheat like we have Triticum durum, which is commonly known as macaroni wheat. It is again tetraploid wheat. Then again this tetraploid wheat was crossed with again a wild grass, goat grass, named as Triticum tauschii, represented by genome D. Then when they were crossed, they produce a sterile hybrid, and it leads to, and after the production of the sterile hybrid, there was a chromosome doubling with the help of certain chemicals like we have colchicine, which induces meiotic errors. So that leads to chromosome doubling and leads to production of this modern day wheat, which is hexaploid, having chromosome number that is 6n is equal to 42. Hexaploid wheat, chromosome number with 42, and this is the common bread wheat. So then comes next question. Edible part of apple is, our options are fleshy thalamus, fleshy endocarp, mesocarp, or cotyledons. As we know that fruit is ripened ovary. Ripened ovary is known as fruit. So This is the simple definition of fruit. And seed is ripened ovule. Ripened ovule. So in case of apple, we don't eat this ripened ovary. We eat the fleshy part, which is known as thalamus. So this one is thalamus. So this is a false fruit. This is a pseudo fruit, pseudo carb. So we eat fleshy thalamus in case of apple. So apple is actually a false fruit, the main edible part of the plant. Apple is actually juicy receptacle, or we can say thalamus or torus of the apple, thalamus. The other examples are peduncle and cashew nut, jackfruit and pineapple. We have some fruits in which we eat certain parts, like we have banana. In case of banana, the edible part is mesocarp and endocarp. Coconut palm, we eat endosperm. Then we have date palm, that is pericarp. In mango, we consume mesocarp, which is the edible part. Then lychee, this is most important. In case of lychee, the edible part is fleshy aril, which is considered as the third integument also. So fleshy aril हम इसमें खाते हैं. In case of Texas baccata, the reddish part which looks like a fruit is again aril. Then in grapes, we used to consume placenta and pericarp. In apple, we consume thalamus. In guava, pericarp and thalamus. Cucumber, we consume mesocarp and endocarp. In orange, we eat juicy placental hairs. In case of pear, again, this is fleshy thalamus. Pineapple, receptacle portion, perianth and brackets. These are the edible parts in case of pineapple. Then tomato, placenta and pericarp. Then pea, cotyledons. Wheat, starchy endosperm. Maize, starchy endosperm. Rice, again starchy endosperm. In case of melon, mesocarp, cashew nut, peduncle, and citrus fruit endocarp. But in case of orange, we consume juicy placental hair. So this is a list of some fruits in which we consume a specific part of the fruit. Next question is, in India, DNA fingerprinting developed at. Our options are National Institute of Immunology, Delhi, New Delhi, then Center for Cellular and Molecular Biology, Hyderabad, Central Drug Research Institute, Lucknow, or Indian Institute of Sciences, Bangalore. So its right answer is CCMB, Hyderabad. So this is the institute where this technique of DNA fingerprinting was developed. And the father of this technique in India is Mr. Lalji Singh. He was considered as father of Indian DNA fingerprinting, Mr. Lalji Singh. So DNA fingerprinting pioneered by Lalji Singh in CCMB, Hyderabad, aids criminal investigations, paternity establishments, and used to confirm identities include Rajiv Gandhi assassins. Singh's work helped solve over 60 legal cases. Proposal for DNA fingerprinting and diagnostic center faces bureaucratic hurdles. So this was the institute where this technique of DNA fingerprinting was developed. And this is a photograph of Dr. Lalji Singh. Next question is, most durable wood among the following is. Our options are Shorea robusta, Cedrus deodara, Delbergia siso, and Tectona grandis. So the most durable wood among these plants is of Tectona grandis. In case of tropical regions or in temperate region, this wood is most durable. So this is the right answer, that is Tectona grandis, commonly known as teak. Its scientific name is Tectona grandis, common name is teak. It is also known by the name sagwan, sagun, and it belongs to family Verbenaceae. And the plant height may reach up to 40 meters in height, and the hardwood is mostly used for making furniture, boat decks, indoor flooring, and is used to make house doors, windows, and is resistant to attack of termites because it contains some scented oils which acts as repellents to the insects. So this Tectona grandis is the most durable wood and is mostly used for making buildings or boats or indoor flooring, etc. Next we have Delbergia siso, commonly known as shisham. It is also known by the name Indian redwood. In Punjab it is known by the name tali. It belongs to family Fabaceae, that is the legume family, and the tree height may reach up to 25 meters. Again, the hardwood is used for making furniture, and it has distinct scent. That is the characteristic of most of the rosewood plant, and it mostly planted on the roadsides and used as a shade tree, and it is also used for making plyboards, musical instruments, furniture, flooring, boat building, carving, etc. Next, we have Shoya robusta, commonly known as Sal or Safed Dummer. It belongs to family Dipterocarpaceae, and the plant height may reach up to 35 meters. And again, hardwood is used, that is dark reddish brown in color, and is very durable and highly resistant to termite attack. It is also used as a fuel wood, and it is easy to saw, easy to cut because of its high resin content. It is difficult to plane and turn. The next, which of the following country leads in the production of natural rubber? So our options are India, China, Malaysia, Russia. So its right answer is China, according to this statistics in 2020. So China stands in third position. The first position is gained by Thailand in terms of production of natural rubber, and India is on fifth position, followed by Malaysia, Cambodia, Myanmar, Philippines, Sri Lanka. Next question, how many carbon atoms are removed in each cycle of beta oxidation? Our options are 1 carbon, 2 carbon, 3 carbon, or 4 carbon. So its right answer is 2 carbon. So long chain of fatty acids are broken down by this process, that is known as beta oxidation, and in every step there is one compound named as acetyl coenzyme A that enters inside the Krebs cycle. So there is a 2 carbon compound which is produced in each step of beta oxidation. So this is the compound, that is acetyl coenzyme A, which contains 2 carbon. And the enzymes which are mostly used in beta oxidation are acetyl coenzyme dehydrogenase. Then we have enoyl coenzyme hydratase. In addition to this, we have this enzyme, that is 3-hydroxyacyl coenzyme dehydrogenase and 3-ketoacyl coenzyme thiolase. So these are the enzymes which are mostly used in this oxidation of fats, long chain of fatty acids, and in every step this 2 carbon compound, that is acetyl coenzyme, which is produced, and it enters in the Krebs cycle for further oxidation of this acetyl coenzyme. The next question is, enzymes of pentose phosphate pathway or of aerobic respiration are found in our options are cytosol of endoplasmic reticulum, mitochondria, and cytosol or mitochondria. As we know that this pentose phosphate pathway is also known as HMP pathway, that is hexose monophosphate pathway, or it is also known as shunt. It occurs parallel to the glycolysis, and the site of occurrence of glycolysis is cytosol. It also occurs in cytosol. In humans, it is very common in the cells of liver cells, and is also found in mammary gland cells and adrenal cortex. While in case of plants, most of the steps occur in clusters. So if we talk about this glycolysis, the first stable product of this pentose phosphate pathway is 6-phosphogluconate. So that's why this is also known as phosphogluconate pathway. And it is mainly used for production of this NADPH and 5-carbon sugars, which include pentose is like ribulose 5-phosphate and ribose 5-phosphate. It is mainly used for preparation of nucleotides, nucleic acids, while this xylulose is mainly used for production of lignin in plants. And this erythrose, that is a 4-carbon sugar, it is utilized for formation of many amino acids. So this is also very important to maintain the oxidative balance or redox balance during oxidative stress conditions. So this is we can say anabolic replicate of glycolysis. Next question is, the first 4-carbon compound found in Krebs cycle is, our options are malic acid, succinic acid, oxaloacetic acid, or fumaric acid. So its right answer is succinic acid, and the first stable compound formed in Krebs cycle is citric acid. And this cycle is also known as TCA cycle, that is tricarboxylic acid cycle, because it contains 3 carboxylic group, and is formed by the combination or condensation of oxaloacetic acid with acetyl coenzyme A, which is a 2-carbon compound. So it leads to production of the 6-carbon compound, that is citric acid. And after decarboxylation reactions, like this is the first acceptor of this acetyl coenzyme, that is oxaloacetate, and acetyl coenzyme, this is a 4+2 carbon that will form this compound, that is 6-carbon compound, that is known as citrate or citric acid, which is formed after condensation. Enzyme used is citrate, synthase, then it undergoes isomerization reaction that leads to formation of isocitrate, and the enzyme used is aconitase. And after the formation of this isocitrate, which is the isomeric form of citric acid, it undergoes oxidative decarboxylation that results in formation of this compound, which is a 5-carbon compound, alpha ketoglutarate, by the release of this carbon dioxide. And after that, it will undergo again decarboxylation and dehydrogenation reaction that leads to formation of this first 4-carbon compound, that is succinate, which is a 4-carbon compound. And after that, there is one enzyme which is used in, that is succinate dehydrogenase. This is the only enzyme which is found in inner mitochondrial membrane, and all the enzymes are found in mitochondrial matrix. Then it undergoes the dehydrogenation reaction again, and here FADH2 is produced, and it results in formation of another 4-carbon compound, is fumarate. Then it will undergo hydration reaction that leads to formation of malate, and finally again dehydrogenation reaction that leads to formation of oxaloacetic acid. So this is how this scrap cycle takes place, and the first 4-carbon compound found in this cycle is succinate or succinic acid. The next question is the nitrogenase converts. Our options are nitrogen into ammonia, ammonia into nitrate, nitrite into nitrate, or nitrate into nitrogen. So its right answer is nitrogen into ammonia. So this is the enzyme which is nitrogenase enzymes, which is made up of molybdenum and iron complexes. It requires a lot of energy, that is 16 ATP, for the production of this ammonia from nitrogen. So this is the role of this enzyme, that is it converts nitrogen into ammonia. Next question is, which of the following enzyme is not proteinaceous? Our options are isozyme, trypsin, holoenzyme, or ribozyme. So its right answer is ribozyme. It is simply made up of, this enzyme is made up of RNA. So most of the enzymes are proteins. Non-proteinaceous enzymes include ribozymes, and it works in aqueous solution, mild pH and temperature, because they are very specific. Enzymes are highly specific. We have examples like proteases, hydrolysis of peptide. It is mainly involved in breakdown of proteins, and it breaks the bond between amino acids and producing free amino acids. So these are proteinaceous enzymes, but this ribozyme is non-proteinaceous enzyme. The next question is, which type of chromatography is used for structure analysis? Our options are paper chromatography, partition chromatography, affinity chromatography, or column chromatography. So its right answer is paper chromatography. In this type of chromatography, it is mostly used to separate the color compounds, and it consists of 3 phases, that is mobile phase, a stationary phase, and the sample. And the separation occurs due to difference in solubility between mobile and stationary phase. So it involves this paper strip, and there is separation of different pigments on the basis of its solubility in, and its difference in solubility in mobile and stationary phase. So this paper chromatography is an analytical method which is used to separate and identify color, chemical, or substances. And for the setup of this chromatography, we require 3 main components, that is mobile phase, a solution that travels up in the stationary phase, and a stationary phase, that is the polar water molecules held in the paper, and the sample. So it consists of 3 components, that is mobile phase, stationary phase, and the sample, and separation occurs due to difference in the solubility and polarity of the sample components as they partition between the mobile and stationary phase during the capillary reaction of the paper. There are different modes of paper chromatography, which include ascending paper chromatography, descending, radial, 2-dimensional chromatography. And this paper chromatography is used for structural analysis of organic compounds, as it separates and identifies components of complex mixture, like we have amino acids, pigments, and DNA bases. So this is used for separation of these compounds, and it's mostly used for structural analysis of these compounds. Next question is, the term bioinformatics was coined by, options are J.D. Watson, Margaret Dayoff, Frederick Sanger, Pauline Hodgeweg. So its right answer is this Pauline Hodgeweg. So she was a Dutch theoretical biologist and complex system researcher studying biological system and dynamic information process. She, together with Ben Hesper, coined the term bioinformatics in 1978 as the study of informatic processes in biotic system. And this bioinformatics is an interdisciplinary field of science for understanding biological data and its interdisciplinary approach combines biology, computer science, statistics, and other disciplines. And its major applications are utilized in analyzing genetic basis of disease, identifying genes and single nucleotide polymorphisms, studying protein sequences, and it is mostly used for identifying these things. And tools and techniques, it mostly used computer simulation, text mining, ontology, structural modeling. So this is all about this bioinformatics. Next question, which of the following nucleic acid has a left-handed helix? Our options are aDNA, bDNA, cDNA, or zDNA. So this is the DNA which shows this left-handed helix. This is the zDNA, and this most commonly found DNA is the bDNA, which is right-handedly coiled DNA. So this looks like a different to the right-handed version. Rather than a smooth corkscrew, the backbone forms a zigzag shape, giving rise to its name. Using an antibody probe, we can recognize that DNA. They found stretches in plasmids, small circular DNA molecules वहाँ पाए जाते हैं, and it base pairs have repeated alternating purines and pyrimidine motifs, particularly the purine guanine and pyrimidine cytosine. The synthetic DNA polymer poly-D, that is GC, poly-D, GC, has been studied by X-ray diffraction of orientation fibers, in which it has either the well-known A or B right-handed conformations or a new left-handed helical conformation. So the left-handed DNA is that DNA, and the most commonly found DNA is the pDNA, which is right-handedly coiled DNA. The next question is, shiny allogeneic sequences are found on. Our options are mRNA, tRNA, small subunit of ribosome, large subunit of ribosome. So its right answer is mRNA. So these are 7 to 8 base pairs from starting codon. These are sequence of bases which are known as shiny sequences, and they act as an initiating site for the attachment of smaller subunit of ribosome. It means जो ribosome के smaller subunit ribosome के complementary sequences होते हैं, they are found in this shiny sequences. So these are the complementary sequences of smaller subunit of ribosome, that is 16S RNA, which is found mostly in prokaryotic mRNA. This is mostly found in prokaryotic mRNA, so it acts as an initiating site for the attachment of smaller subunit of ribosome. So shiny sequences are found on bacterial messenger RNA and plays a crucial role in facilitating the initiation of protein synthesis by aligning the ribosome with the start codon AUG. Thus, ribosomal binding site is commonly present in bacterial mRNA and rarely in archaeal mRNA. So this is mostly found in prokaryotic mRNA. So this was all about for today's discussion about these questions. I hope you will get some idea from this presentation. If you have any questions, queries, you can ask in the comment section. Thanks for watching. Have a nice day.